myxomavirus evolution of the Toledo L3 variant and the surveillance of genes implicated on the species jump. I'm going to introduce you to the disease itself, the virus, different cell lines that I use, the methods, my results. We'll discuss them a bit, and then we will conclude this presentation. So to introduce you to this virus, this is a lepropox virus native to South America, where it infects a rabbit. It does not present serious symptoms. However, when the virus got introduced to Europe in the 50s as a measure to control wild European rabbit populations, it changed the symptomatology. The infection went to 9 to 12 days with fibromas or myxoma showing up on the ears, eyes, and genitalia of the rabbits. There's also purulent discharge from the eyes and nose, which ends up usually being a fatal disease. However, a few years after its introduction, something very interesting happened where genomic mutations gave rise to attenuated strains. Once these strains developed, the genome remained stable. That is until 2018, when there was a species jump discovered. Now, to understand this species jump, I'm first going to tell you a little bit about what the genome of classical myxoma looks like. This is a fairly large genome composed of 161 kilobase pairs that encode for 171 open reading frames with over 100 genes ranging from M000.5 to 1 M156. Now, with this species jump, it changed host. The new host is the Iberian hare, and here you see a picture of the whole genome. Each arrow represents a different open reading frame, and there were 3 key mutations detected. The first one was an insertion of 2.8 kilobase pairs into gene M009. There's also a truncation of gene M036 due to the addition of 4 adenines, and a truncation of gene M152 due to the insertion of a cytosine. So now I'm going to explain to you a little more about the mutation seen on gene 009. So this insert right here contains 4 genes that are homologous to either different pox viruses or to the myxomavirus itself, this cassette right here, which will be homologous for genes 60, 61, 64, and 65. The interesting feature of this cassette, however, is that it's inverted in nature. These mutations gave rise to several questions for us. So how is this virus going to evolve in nature? Because the first time that it did a species jump here in Europe, it actually developed into several strains. So is that genome going to remain stable right now, or is it going to mutate? And what about the mutations that we have already seen on different genes? Are they actually essential for this species jump as a whole, or is it only one of them? So we set out a few objectives to better understand this. The first was the study in vitro of the evolution experiment with the hair myxoma virus. This will probably help us see more cytopathic effect changes and how it looked like in the wild. But also we wanted to survey the genes that I just explained to you, both from in vitro samples that I would acquire here and from in vivo samples that were acquired from the lab. So onto the cell lines. To do these experiments, we needed models that were representative of both the previous and the new host of this virus. The new host, which is the hair, is represented by the HNR cell line. And then we have the rabbit cell line, the RK13 cell line, representing the previous host, which is the rabbit. The virus that this thesis is focused on is actually the hair myxoma virus, an isolate named hair myxoma Toledo 18L3 virus with a titer of 1 × 10⁵ plaque-forming units per milliliter. An interesting fact about this virus is that its tropism is able to infect not only the hair cell line, but also the rabbit cell line. So we'll be studying that in parallel. We also compare with the Lausanne myxoma virus, which is a classical strain of myxoma that has a titer of 1 × 10⁶ plaque-forming units. The Lausanne virus has a tropism that is only able to infect the rabbit cell line. It cannot infect any other leprids. Now for the methodology. One of the biggest experiments that were performed was an evolution experiment done in parallel on rabbit and hares. The image here depicts what was done for simplicity purposes only to the hair cell line. So we will start with a low infection mode to prevent the virus from developing any diffusion variants if you go to a higher mode. So we'll infect and allow the infection to go for 72 hours, at which point it was harvested. This period of time is defined as a passage, and this experiment went on for 15 passages in total. Every so often, the viral titer was also assayed. We will also see with this the growth curve, plaque morphology and size, and we'll do some DNA surveillance on this experiment as well as in vivo samples. Now, on to our results. As I mentioned previously, the evolution experiments seek to force the evolution in parallel on two cell lines of this virus. These will allow us to monitor the cytopathic effect, or CPE, see the viral titers changes, as well as how the mutations progress over time. Here, you see one of the initial passages that were done. On top, you see the control cell lines, which were not infected, and then the infected cell lines with the same hair myxoma virus. Throughout this presentation, you will see a code like this, where the first two letters represent the cell line that was grown. Then the virus is represented by L3, and then the passage number will continue to change as the experiment progresses. So, pointed with arrows, we see classical myxoma formations on the cell lines. In circles, we also see areas where the monolayer lifted, either due to infection itself or the health of the cells, but we also see some myxomas. Now, the myxomas are 3D structures that usually resemble tumors, and we know that these are the classical myxoma-like appearance because we also infected the rabbit cell line with the Lausanne or classical myxoma to compare, and we see those same 3D structures popping out. A very interesting result, however, was achieved at passage number 6, where for the virus grown on hares, the plaque size actually seemed to get larger, and on the virus that had been growing on rabbit cells, we see a new cytopathic effect. This is called a syncytia, which is usually a multinucleate cell resulting from the infection of one single cell that then is affected by the fusion proteins that are expressed by the virus, and their interaction with the host cause this cell membrane to actually fuse with the neighboring cells, spreading the infection. And it can be composed by several hundreds or thousands of cells. At this point, we weren't quite sure if this was actually an effect of the virus, because it is not a common myxoma effect, although it's a common viral side effect. So we continue the experiment, and on passage 15, we see that the myxomas that have been resulting from the virus growing on hair cells are still very defined and uniform. They're very easy to see. Whereas we see another change in CPE for the virus that has been growing on rabbits. We now see a mixture of myxomas with syncytia. So we know something molecularly changed here, but we don't know if this CPE is caused by the host virus interactions, and if it will change if we grow this virus on the hair cells. So, to study this, we then infected the hair cell line with different passages from the virus grown in different hosts. For the most part, we saw the appearance of myxomas, which was the expected CPE, but for the later passage of the virus grown on rabbit, we saw syncytia alongside myxomas as well. So this CPE is conserved from now on. And another part that was going to be studied on this evolution experiment was actually the changes happening to genes 009, 36, and 152. We chose three representative passages, which were passage 5, 10, and 14, to screen for these genes. For gene 009, as we can see here, we see in this band that is almost at 3,000 kilobase pairs that it must contain that region of 2.8 kilobase pairs inserted. Whereas when you screen the Lausanne or classical myxoma for that same region, it's barely reaching 500 base pairs. Therefore, these are positive hair myxomas, at least for the mutation seen before on 009. Sequencing analysis was done for 152 and 036. The cytosine was conserved in all passages, and no new mutations were detected on gene M036. One of the other things that needed to be done with this study was to see how the viral titer changed from all 15 passages. Now, most of this experiment was done blindly, hoping to keep the mode of infection low. However, when you retroactively take the viral titer of the virus grown on rabbits and the virus grown on hares, you see that it changed from passage to passage, but overall the trend is that it grew for both of them, with the titers from the virus growing on rabbit cells usually being higher than those for hair. This is, however, a snapshot of only one point. So we wanted to see how it would look like if we did a growth curve. Growth curves are usually done in triplicate. So what will be done is we will infect different cell cultures with different passages, the initial passages and later passages of this virus. The growth curve allows to see how this virus replication rate change over time. Now, usually, growth curves for virus actually peak at 72 hours, but the interesting result that we see is that for the latest passage of the virus grown on hares, it actually extended all the way to 96 hours. This suggests that the virus has been adapting to its new host, but we don't know if this growth curve is larger than this, as we stop the experiment at 96 hours, but it will be interesting to continue this growth curve to see how far it goes. Now, these viral titers have all been done on the RK13 cell line, so we weren't quite sure how they will compare if we did them on the hair cell line, where they will grow naturally. So the general trend that we see when we assay the first and later passages is that the viral titer increase over time. And then when you grow it on the hair cell, you'll see the same trend being true. However, the passage does decrease in titer when it's grown on the hair cell line than on the rabbit cell line. We saw different CPE changes, one of them being that the hair's plaque assays were getting larger. So we wanted to see how these actually measure with numbers. So we took the area of several plaques and counted them, averaged them, and noticed that indeed the area of the plaques has been getting larger, especially when it grows on the hair cell line than when it grows on the rabbit cell line. And you can see it here on these pictures, the difference of the same virus growing on two cell lines. We also wanted to see what happened to the virus that had been growing on the rabbits. So we see the opposite. The plaque size actually decreased over time, and we believe that this might be due to the appearance of syncytia on passage 6 that carried through the whole experiment. So we infected the virus and counted the amount of nuclei on the syncytia. We found then that the syncytia that was growing from the virus on the rabbit cell lines was twice as large as that from the hair cell line. However, this result may be a little underestimated, as it was very hard to see the nuclei from the light microscope. Now, we're going to move on to the surveillance of in vivo samples for the genes 009 and 152 as well as 36. So the lab have been acquiring samples since the year 1992 to 2020, both from rabbit and hares. I assayed 71 of them, 31 from hares, 44 rabbit, and I know from personal communication with my tutor that all of these samples had been previously screened for gene M009. All rabbit samples did not contain that addition of 2.8 kilobase pairs, and all hair samples did. So I focus more on gene 36 which is a very large gene. It's about 3,000 kilobase pairs. And here you see a compilation of different mutations that were seen on this gene. It's very interesting to see that most of them were actually insertions or deletions, which don't tend to happen often. So one of such mutations, when you compare the classical myxoma sequence to the hair myxoma sequence, is that addition of 4 adenines that I mentioned earlier, highlighted in green. But what I saw was the addition of a 5th adenine, which I highlighted in red. And this was in several samples. Another mutation that was interesting was on a region that is usually conserved between the hair myxoma and the classical myxoma. What I saw was either a deletion of adenines on this group of 2, or the deletion of an adenine on a group of 4, or the deletion on both regions. This suggests that this gene actually has great plasticity and might not be of use for this virus. Now on to gene 152. Gene 152, as we know, got truncated. It usually codes for a serpin, which is an immunosuppressor. By it being truncated, it might be what is aiding on the infection of the hair. Now, but we had 71 samples and needed a high throughput assay to study all of this. So we took advantage of an assay that was being developed by the lab. The way that I optimized this assay was first by confirming that there were samples that contained the 2.8 kilobase per insert on gene M009 to determine which were hair and which were rabbit. From then, I sequenced those same samples to confirm the addition of the cytosine on the ones that I determined had the insertion. And this actually allowed us to put to use a Mel curve analysis. This Mel curve analysis will give us a lower temperature for the rabbit samples, which had no cytosine, or the extra cytosine, I should say. And then the hair samples will have a higher Mel temperature. Now, this assay still gave me a few mixed results that later I had to sequence to confirm identities. But overall, I found that all 31 hair samples did contain that additional cytosine, and none of the rabbit samples had it. As a summary of what I did for this master thesis, well, my in vitro evolution experiment unveiled different changes in CPE, the viral titers, plaque morphology. I also found that the hair myxoma Toledo 18L3 virus had adapted better to grow for the hair cell line. The mutations that had been previously described on gene M009 and M152 have been conserved in all in vivo and in vitro samples, and the large number of mutations found on gene M036 showed the great plasticity of this gene, and that is no longer essential for this virus. Now, with your permission, I will read my conclusions. The in vitro evolution experiment led to a change in CPE characteristics of myxoma, which affects virus titer. Morphological changes seen in the CPE of RKL3 indicate lower viral titers, which could in turn lead to longer lasting infections. Changes in plaque size did not produce considerable changes in replication rates. The number of cell-forming syncytia is approximately 5 times larger than on a viral plate. RKL3P14 syncytia grown in RK13 cells is twice as large than when it is grown in HNR cells. The virus titer of HNL3P14 was higher than RKL3P14,